Greeting everyone. In today's session, we will explore the multifaceted role of brassinosterides, an emerging player in plant growth that exerts a potent influence on various aspects of plant development. Brassinosterides are gaining recognition for their diverse and pivotal roles in shaping the growth and overall development of plant. So, let us start the new and emerging hormone. Brassinosterides are a class of plant hormones that play a crucial role in various physiological processes including growth, development and response to stress. The discovery of brassinosterides dates back to the early 1970s when scientists were investigating the role of pollen in plant growth. Here are the key milestones in the discovery of brassinosterides. Mitchell and colleagues conducted a screening of pollen from approximately 60 species and around 30 of them were found to induce growth in bean seedlings. The substance responsible for promoting this growth was named brassins. In 1974, the quest for the active component of brassinosterides was collaboratively undertaken by scientists from USDA, that is United States Department of Agriculture, USA, operating at the Northern Regional Research Center in Pretoria, the Eastern Regional Research Center in Philadelphia and the West Valley Agriculture Research Center in Maryland. A quantity of 500 pounds of pollen collected by bees underwent a solvent extraction processes using 2-propanol at the ERRC. Subsequent to this, at the bark, there was successful partial purification of the extracted material. However, at the NRRC, the substance underwent crystallization and was subsequently analyzed using X-ray techniques to determine its molecular structure. This steroidal lactone, identified as a biologically active plant growth promoter with the molecular formula of C28H48O6, was officially named brassinolide. In their research conducted in 1979, Grove and his colleague successfully isolated this compound from the pollen of the rapeseed plant called Brassica napus, contributing valuable insights into the understanding of brassinolide and its significance in plant physiology. As of available information, a total of 70 brassinosterides have been isolated from 64 different plant species. Among these, 5 are conjugated, 65 are unconjugated, while 5 are conjugated forms. BRs exhibit a widespread presence across various plant groups, highlighting their significance in the regulation of diverse physiological processes. In angiosperms comprising 12 monocotyledons and 41 dicotyledons, while gymnosperms represented by 6 conifers also contain brassinosterides, indicating their conservation across different seed-bearing plants. Pteridophytes such as Equisetum arvens showcase the presence of brachiosteroids in vascular plants that reproduce through spores. Even in bryophytes like Mercantia polymorpha, the existence of brachiosteroids underscores their involvement in the life cycle of mosses. Furthermore, algae including Hydrodictyon reticulatum, Coleura vulgaris and Cytoceras marica demonstrate that these hormones are not confined to land plant but extend to various aquatic photosynthetic organisms. This widespread distribution emphasizes the evolutionary and functional importance of brachiosteroids in the plant kingdom. This diversity in brachiosteroid structure and sources highlights the complexity and versatility of these plant hormones across various plant species. Researchers continue to explore and identify new brachiosteroids, contributing to a deeper understanding of their roles in plant physiology and potential applications in agriculture. As now you know, the presence of brachiosteroid is widespread in the plant kingdom, encompassing both higher and lower plant species. It can be detected in different plant organs such as pollen, anthers, seeds, leaves, stems, roots, flowers and grains. Notably, higher concentrations of brachiosteroids are observed in young and actively growing tissues in comparison to mature tissues. Brachiosteroids are notably present in high concentrations in pollen and immature seeds ranging from 1 to 100 nanogram per gram fresh weight. In contrast, shoots and leaves generally exhibit lower levels of BRs, typically ranging from 0.01 to 0.1 nanogram per gram fresh weight. Cupressus arizonica stands out with the highest concentration of brachiosteroid, so causing a significant 6.4 milligram per 1 kilogram of pollen. This slide shows the transport of brachiosteroid. So, unlike other plant hormones, they do not undergo long distance transport. Instead, they exert their influence in a paracrine manner, acting on the neighboring cells at the locations where they are synthesized. In the paracrine functions, the hormone is produced and binds to its receptor in nearby cell, influencing their function. So, this slide is about the biosynthesis of brachionolyte, a type of brachiosteroids which involves several enzymatic steps within plant cells. So, let us discuss it in detail. Brachiosteroids are synthesized from the sterol, that is campesterol, a compound structurally akin to cholesterol. Enzymes belonging to the cytochrome 450 monooxygenase family associated with the endoplasmic reticulum catalyze the majority of reactions within the brachiosteroid biosynthesis pathway. As illustrated in the figure, The levels of bioactive steroids are intricately regulated by various inactivation or catabolic processes encompassing epimerization, oxidation, hydroxylation, sulfonation and conjugation to glucose or lipids. Despite these complexities, the identification of enzymes responsible for steroid catabolism and inactivation remains limited, with only a few such enzymes currently identified. Moreover, active steroid levels undergo a regulation through negative feedback mechanism dependent on steroids. When hormone concentrations surpass a specific threshold, there is a reduction in steroid biosynthesis. This reduction is achieved by the downregulation of genes involved in steroid biosynthesis and the upregulation of genes associated with steroid catabolism. Consequently, mutants with impaired responsiveness to brestanolide tend to accumulate elevated levels of active steroids compared to wild type plants. This slide describes the physiological role of steroids. As you know that steroids regulate several physiological processes in plants such as seed germination, flowering, root growth, senescence, cell division, ATPase activity, increase in the nucleic acid content, that is DNA or RNA. Now we will discuss each physiological response separately regulated by the steroids. Coming to first physiological role, which is stimulated by steroids, is seed germination. Application of brestanolide to seeds of Lepidium sativus and eucalyptus has been documented to enhance the percentage of germination. Likewise, other steroids like epibrastinolide and 28-homobrastinolide have demonstrated the ability to increase germination percentage in various plant species including brassica napus, rice, wheat, tomato, tobacco, groundnut, vigna, etc. The application of steroids to the foliage resulted in an increased number of flowers in strawberries. However, for grapes, the effect of steroids on flowering varied based on the timing of application. Specifically, the applying steroids during autumn improved the number of flowers, but if the application was delayed until later winter, it had an inhibitory effect on flowering in grapes. The next one is the application and increase or decrease in the root growth. When it comes to root growth, steroids exhibit a dual role, influencing both promotion and inhibition. The impact on root growth depends on the concentration of steroids, with low concentration promoting growth and high concentration inhibiting it. Now let us discuss the senescence. The application of brestanolide induces senescence in xanthium and Reumax X plants. Additionally, steroids accelerate the senescence process in detached cotyledons of cucumber seedlings and in the leaves of mung bean seedling. Notably, Arabidopsis mutans deficient in Bresnostride display a postponement in the senescence of chloroplast. The presence of Bresnosteroids led to an increased rate of cell division. The stimulatory effect of Bresnosteroids and cell division have been observed in various systems, including cultured parenchyma cells of Helianthus tuberosus. In this particular system, the application of nanomolar concentrations of Bresnosteroids resulted in a significant increase of at least 50% in the rate of cell division. Bresnosteroids also regulate the activity of plasma membrane ATPase. When root membranes were exposed to Bresnosteroid, there was no notable increase in the activity of ATPase. However, in the presence of the reaction mixture containing epibresnoloid and IEA, the ATPase activity was higher compared to treatment with IEA alone. These findings suggest a potential synergistic effect between epibresnoloid and IEA concerning ATPase activity. Bresnosteroids play important role in increasing nucleic acid content. It significantly increases the content of DNA and RNA along with enhancing the activity of RNA polymerase in mung bean epicortical tissue. Similar enhancement in RNA and DNA content were observed in mustard, wheat and tomato upon the application of BR. So, now move up to the molecular mechanism of action of Bresnosteroids. The molecular mechanism of action underlying the promotion of cell elongation by Bresnosteroids involves a complex signaling pathway. Let us discuss about in detail. The plant cell wall constitutes a tightly interconnected and firm matrix that hinders both cell expansion and differentiation. For elongation and various morphogenetic processes to take place, you all know there is a need for modification to the cell wall. This involves processes such as wall relaxation or loosening, along with the incorporation of new polymers into the expanding wall. These modifications are essential to uphold the integrity of the cell wall during the growth and development of the plant cell. Numerous proteins implicated in the processes of cell wall modification have been discovered, including glucanases, xyloglucan, endotransglycosylases or Zs, and expansion. Although the intricate nature of the cell wall was made it challenging to establish a definitive molecular model for wall expansion. A probable scenario proposed by Cosgrove et al., which suggests that expansion play a primary role in wall relaxation. However, the activities of gluconeases and Zs can influence expansion activity by modifying the viscosity of the hemicellulose matrix. Additionally, Zs might contribute to the integration of new xyloglucan into the growing wall, and cellulose biosynthesis is anticipated. Nonetheless, it is confirmed that breast induce changes in the biophysical characteristics of plant cell walls and elevate the abundance of mRNA transcripts associated with wall modifying proteins including Zs. In the process of elongation in soybean epicotyls, the application of breast resulted in a rapid increase in the plastic extensibility of the cell walls within a span of 2 hours. This coincided with a rise in the mRNA levels of a gene known as BRU1, displaying significant similarity to various xyloglucan endotransglycolases. Subsequent enzyme assay using recombinant protein confirmed the functional nature of BRU1 as an Z. Furthermore, a proportional rise in extractable Z activity in the epicotyls was observed with increasing concentrations of applied BR during the early stages of elongation. Notably, the BRU1 gene exhibited a specific regulation of BRs during the early elongation stage, and the increased expression was not only or solely a result of enhanced elongation. Consequently, BRU1 is likely to play a crucial role in the BR promoted elongation of soybean epicotyls. However, the role of expansion in this particular system has yet to be explored. So, this lecture is coming to end. The next part of this plant hormone, that is breast signal transduction, will continue in the next lecture.